Hi all, thank you for joining. This is the first of an annual seminar series titled the Pendleton Science Round-up. And this event is hosted by USDA Ag Research Service and Oregon State University's Columbia Basin Ag Research Center in collaboration with Wheat Talks of the Oregon Wheat Growers League. And the idea for this seminar series really came out of a conversation that I had with a producer where I recognized a greater opportunity to communicate with our producers and also to have science presented in an open discussion format. So this talk will be about 20 to 25 minutes, and at the end, we'll have plenty of time for discussions and questions and answer sessions. So each year for this seminar series, we're going to invite 3 local, us being Adams, Oregon, and an outside speaker to present on topics that include dryland ag, technology, soil health, and other issues. So we'll take some time and we'll jump on in. So the first of the talk is Farming for Better Soil Health. My name is Kate Reardon. I'm a microbiologist here with the Ag Research Service. And so the first question here is, what is a healthy soil? Well, the NRCS, the Natural Resource Conservation Service, they describe a healthy soil as a soil with the continued capacity to function as a vital living ecosystem that sustains plants, animals, and humans. So important words in there, a vital living ecosystem. And I think that this definition of soil health really pulls it from soil quality, which is more of a function of a soil, a capacity of a soil to function. So different properties of soil, especially for a healthy soil, we'd want to look at the chemical, the physical, and the biological aspects. So for chemical, we think, well, a healthy soil would have an optimal pH, available nutrients, low salinity, and good levels of organic matter. For the physical properties, we look at the soil structure, saying a good soil structure with nice aeration, adequate water storage and drainage, and also presence of soil aggregates. On the biological side, a soil would be diverse. Now, I'm really interested and focused mostly on bacteria and fungi, but it's important to consider also these higher trophic levels. So the nematodes and the microarthropods. But a very biodiverse soil. It also have low pest pressure, a lot of beneficial organisms, whether they suppress plant disease or they cycle nutrients. Here at the bottom is a quote by Noah Fear: "Soil health, like beauty, is in the eye of the beholder." And I really enjoy this quote because soil health isn't just a perfect box. It's a box made for an environment, a box made for a system. So if you're farming in 9 inches of annual rainfall, you may never achieve that 4% of organic matter. And also if you're a blueberry farmer and you need that acidic soil for your crop to grow, that's going to be a very different parameter than, say, a wheat farmer would expect. So thinking about that within that context. Now, how do we measure soil health? Well, there are different indicators. So there are some guidelines on what an indicator should be. You need to measure the chemical, physical, and biological soil properties. So indicators should be related to those. They measure changes in soil function, and that's really important. So measuring changes or trends. It needs to be sensitive to management in the environment. If you're implementing specific managements and your indicator's not sensitive, then you're going to be struggling to be able to track those trends and changes. And also being able, it's easy to measure and accessible to users and applicable to different field conditions. Soil health indicators revolve around, well, there's, I guess, 3 main soil health indicators. I guess it doesn't revolve around those. But organic carbon concentration. So this reflects the nutrient cycling retention, soil structure, available water holding capacity. There's carbon mineralization potential. So 2 of these that are commonly looked at is beta-glucosidase activity, which is a nutrient cycling enzyme, and also soil respiration. These reflect the carbon and nutrient cycling capacity, and they're strongly related to microbial biomass and activity. And finally, aggregate stability. If you were at the direct seed meeting, you might have learned how to measure this yourself on your own soils. So this reflects the resistance of the soil to erosion, whether wind or water, and also the soil infiltration and storage. So the question is if you can measure changes in your soil health, what can you do? You can shape healthy soil. So how do you shape a healthy soil? Well, I show this probably way too much in my talks, is this interaction between the environment and the microbes. So the environment, that is the microbes environment, not necessarily like city versus county. It's going to be the exact and immediate environment around those soil communities. So thinking about the chemistry and the physical properties of those soils, you can shape that environment. And that environment, the chemistry, the pH, the nutrient availability, that's going to influence those soil microbes. The soil microbes' respiration, so what can they consume? How do they grow? How fast do they grow? So by changing that environment, you influence those soil microbes, and then the soil microbes, through their physiology and through their respiration and metabolism, they then change that environment. So we have the capacity to change that environment and therefore influence and change those communities. So NRCS has 4 soil health planning principles. They say minimize disturbance, maximize biodiversity, maximize soil cover, and maximize living roots. I've also added recognizing inputs. There's a lot of research that goes into how what you do or what you apply changes the soil communities. And so I've added this here on this planning principle. So let's zoom out a ways. So if we say that we can change the environment, let's look across environments, across different climates on different precipitation, different soils. And this is a study by Veronica Costa Martinez, where she looked at 15 different locations at field research experiments to try to identify how farm management affects soil biology and soil health indicators. So these field experiments were not coordinated. They varied based on the different regional cropping systems, whether it was going to be cropping with wheat, corn, millet, rotations, annual crops, fallow, had lots of different tillage treatments, different variations in tillage with the intensity and the frequency, and also whether it was irrigated or dry land. What they did was they tried to take all of these field experiments and be able to identify whether conservation practices, which they indicated as reduced tillage, cover cropping, crop diversity, or manure application, whether these practices change the soil health indicators or influence the biological communities. So to do this, they used a response ratio. So in every experiment, they had a treatment being that conservation practice and what would be considered a standard. So you'll see those conservation practices up here along the top bar. And along the bottom is this response ratio. Now this axis will change, so sometimes they look like the same, but it's going to be a much larger effect. When they took these, they compared and saw how different the standard was from the conservation, and they used that to normalize the data. You'll see n equals something, and that number means how many different comparisons were in this data set. They looked at organic carbon, so one of the soil health indicators, microbial biomass, and they did that using fatty acid methyl esters, so the fatty acids in the microbial biomass from the soil. Using that method, they can break down and look at different soil groups. So gram-negative bacteria, these are organisms that can consume rapidly, easy to use substrates. Gram-positive bacteria and fungi break down more recalcitrant substrates or organic matter. Actinobacteria, these are really cool gram-positive bacteria because they look like fungi. They make the big filaments. And they're known for disease suppression and nutrient cycling, as well as antibiotic production. Saprophytic fungi, great decomposers, and arbuscular mycorrhizal fungi, which associate with the roots. So when you look at these graphs, it'll be an orange bar, and anything to the right is a positive effect. When they looked at reduced tillage, this was within 15 sites, you can see that everything was shifted to the right, meaning there was a positive effect except for saprophytic fungi. Cover cropping had a positive effect on the organic matter and all of these microbial groups, with a stronger effect here on the arbuscular mycorrhizal fungi. And with crop diversity, this was interesting because the more crops they had, or the more intense rotation, didn't have any effect on carbon, microbial biomass, or any of the groups except for the arbuscular mycorrhizal fungi, which had a lot of variability from a marginal effect to a very significant effect here. Manure application had broad and significant effects on all of those components. So by changing that environment, changing how they manage the soil with a disturbance or keeping the ground covered or adding manure, they were able to change these communities and the abundance of the microbiology and microbes in those soils. So question is if you were going to do a different practice or you wanted to implement a different method, does it matter in the long term, or is this something you have to keep up with? So I piggybacked on a study with Stuart Wiest and Harold Galani, where they were looking at the effects of different surface amendments on soil carbon. And here in this picture on the left, this was conducted under no tillage in 16 to 18 inches of annual rainfall here in the station in Adams. They grew continuous wheat, or they had continuous fallow. And in these two treatments, they did a surface application of several different amendments, all at the same amount of carbon that was applied. They added biosolids, manure, wheat compost, wood sawdust, or sugar in the form of sucrose. Now, I picked just a small subset of these, one, because they were relevant, and two, because I thought they were super cool. And they grew different crops. So they had the annual wheat, or they had an annual brassica crop, the Brassica juncea or Brassica napus. Also grass, tall grass fescue, and fallow, which is not a crop, but I grouped it with the crops. The treatment phase was 5 years. So for 5 years, they grew wheat, or it was fallow, and they applied these surface amendments, or they grew the brassica or the grass. And after that, they homogenized the field. So they took fallow for 3 periods and followed that by wheat for 2 crops. This is the homogenization period, the time when the organic matter would break down. And it was after this point I looked at the soil communities. We looked at the 4-inch depth, so kind of the really nice active zone. Oh, excuse me. So for durability, we wanted to look at organic matter breakdown. So one second. So what I want you to see on this graph is that when organic matter is broken down, it's not a trivial factor. It's a lot of microorganisms contributing a lot of different soil enzymes. Here's cellulose degradation. Cellulose is pretty simple. It's a bunch of glucose molecules chained together. But to break this down, it takes different enzymes. It takes these exoglucanases chomping down from the outsides of the crystalline matter, amorphous crystalline regions where you have other enzymes able to break that down from the center. And then when you get small chains, beta-glucosidase is able to come in and to pop off the glucose. So thinking about this, it's a consortium of those microorganisms that are driving this organic matter degradation and the availability of these nutrients. When we looked at these treatments again for durability 7 years after these amendments, we looked at enzymes related to the sulfur cycle, so releasing sulfate from organic matter, The carbon and nitrogen cycle, this releases amino acid sugars, the N-acetyl-glucosaminidase. This breaks down chitin, so those fungal bodies or insect bodies. Arylamidase, this breaks down peptides to amino acids. It's important in the nitrogen cycle, and it's an index of net soil nitrogen mineralization. For carbon cycling, we looked at the breakdown of cellulose and galactose, for beta-glucosidase and galactosidase, and finally acid phosphatase, so the release of phosphate from organic phosphorus. What's interesting about this, well, first, I guess, is how to read it. So letters that are shared, so A's, are not significantly different. The continuous grass, 7 years after the amendment stopped or it wasn't grown, was significantly greater than any of the other treatments. It had a durable effect on the nutrient cycling activity. It was significantly greater than the other annual crops. It was significantly greater than fallow. And so this is a durable effect that those communities had changed, maybe in the community structure number, but definitely in the community function. And this was a durable effect. But what about the community? So breaking it down to the bacteria and the fungi. When we looked at the bacteria, there was no difference. There were no durable effects. The communities, if they had changed, they had reverted back to the abundance that they were before. They were not distinguishable. And we estimated this based on DNA and on gene copies. When we looked at kind of the who's there and how many, the community structure, the red circle indicates that these treatments are not distinct. After 7 years, there were no durable effects on bacterial community structure. The fungal abundance was definitely interesting because 7 years later, annual crops, that grass treatment here was significantly different than wheat, brassica, or the fallow. And we saw that it was also significantly greater than the manure treatment that was under wheat. We also saw that wood, so when wood sawdust was laid down, that had a durable effect on those communities. And this wasn't really mind-blowing because fungi are able to break down wood. They're really good at breaking down wood, but it's also a harder to break down substrate. And that some of the wood had remained in those plots. So we saw that it is still promoting those fungal communities. But what's really interesting with this is when we looked at the community structure, we saw something unexpected. Here in the red circle, these communities are similar. They're not different. They're not distinguishable. None of these treatments had a significant effect, except if you look here at wood, where we saw the high abundance, we saw that this community was also distinct. It was distinct from all the other treatments. In addition, right down here, hidden at the bottom, was the sucrose treatment. The sucrose treatment produced durable effects that had changed those communities where they never reverted back. And this is really interesting on the sugar because in old research from, I guess, the 60s, when they added sucrose to soil aggregates, they found that it would promote or enhance the fungal numbers more than the bacterial. But we're not sure, and it's really unknown what in that environment, so what that sucrose did specifically to that environment to have such a lasting impact on those fungal communities. So overall, what you do impacts soil biology. We think about when you have the environment here, you can change the environment, you can shape that environment, and that environment influences the soil biology. That soil biology then drives a lot of that soil function, the nutrient cycling, the aggregation, and overall, you can be able to shape and to change that. And sometimes, depending on what you do, with durable, lasting effects. So reflecting back on the 4 pillars of soil health, we saw that disturbance had a positive effect on the soil biology. Biodiversity is in the cover cropping and the soil cover crops did as well. And looking at the data on durability, those maximizing those living roots also had a big impact. So if you're looking at soil health, you can send your soils to different laboratories and they can run several analyses to look at the chemical, the physical, and the biological properties. But NRCS also recommends these in-field observations as well. Soil cover, residue breakdown, look at the water. Do you have water issues? You can measure your water stable aggregates just at home. But what's important is looking at those trends. So not trying to achieve the highest amount of organic matter, but what is improving your soil and is what you're doing trending in a positive way. So I'm going to open for some questions and just general discussion, but I first want to give a shout out for next week's Pendleton Science Round-up seminar by Francisco Calderon. He'll be presenting on long-term soil carbon research at the OSU Columbia Basin Ag Research Center. The third week of February, we have Nicole Durfee with the USDA on hydrology and dryland agriculture, followed by our guest speaker, Tom Ducey, taking control of your soil's health, influence of amendments, biologicals, and management. So with that, I will open up for any questions. I have a question here for Greg Goad. He always asks me, so I have a post-it note just for him. One of the questions that comes up a lot is how many organisms are in a teaspoon of soil? And the estimates are 100 million to a billion bacteria. But there's a new publication out that's looking at the number of species on Earth. And what they're estimating is 430 million species of bacteria, 5.6 million species of fungi, nematodes 470. And I think what's mind-blowing is for phage, so the viruses that attack bacteria, 9.9 billion different phage. So that one's for you, Greg.
So what was the question?
Oh, I guess I didn't have a question for you.
Okay, good.
Yeah.
So I don't think I could answer it anyway.
Yeah. Go ahead, Dave.
Great presentation, Kate.
Oh, you're muted though.
How about now?
Oh, perfect.
Okay. Good to see everybody. Good to see you, Dan. It's been a while. Good to see everybody, though. Let's see. One of the things what you just said about viruses and phage brought me back to my postdoc when I was presenting at the University of Idaho. And I can't remember the professor's name, but it'll come to me probably. A very interesting character. And he said, well, what about all the phage in soil? Because I was talking about soil microbes and the bacteria and the fungi side of things, streptomyces, actinomycetes. And I said, well, we really didn't look at that. And he commented that the mass of phage in seawater far outnumbered or far outmassed everything else, which he, I'm not going to say he was a loon, but he always had some interesting things to say. But what you just shared about the diversity of phage, and we know that they attack bacteria and they do what they do. But that whole interesting community of what's going on there, what is it having effects on agricultural productivity? And that might be an interesting black box to further explore. Probably not easy, because they're phage and they don't grow unless they're on their host. And you got how do you grow bacteria that don't grow and things. Anyway, really liked your presentation. The diversity questions, it's always a question, but then how did that roll forward to affect the productivity or the resiliency of your system. So nice work. Thanks, Kate.
Oh, thank you, Dave. So one thing I always thought that would be fun is kind of delving into phage, and I imagine it's like during The Walking Dead, the whole zombie apocalypse, but these phage come in and just blow up cells. So you have all of this carbon just stuck to the walls from these bacteria just blowing up. I always thought that'd be super fun to look at.
There's a science fiction book called Darwin's Radio that kind of touches on that. A little bit of a freak out story, but anyway, if anybody needs a book to read, it's not bad. It touches on that topic.
Oh, good. Clint?
So can you go back to that screen that shows where they all are clumped together, and explain that a little more?
Sure. On the fungi?
Yeah, the fungal community structure. Yeah.
Okay. So with this analysis, it was terminal restriction, fragment-like polymorphism, but basically what that says is it's not like the sequencing of the communities that we see a lot, or mostly that's all done now. And so it looks at the genetic diversity. So how different are the genes within this fungal community? And it groups them. So if they're clustered together here, like this center, it means that those communities are very similar. The further apart they are, like wood and sugar, these are very different. So the fungi that are there are going to be different than the fungi in here. There's some overlap. It depends on how many there are, and then the diversity. So if you have 3 communities and they all have organism, I don't know, I can't even make that up for you, Clint, but basically this just shows the clustering means are very similar. The further apart they are, the more different those communities are. Does
So
that help?
the wood and the sugar then brought in different communities.
Oh, what's that? Yes, sugar and wood, but this is 7 years after they were added. And I think that that's what's the really cool part of this is they were durable effects. I wouldn't have expected sugar to have a lasting effect, especially since there was no effect on pH, because it looked the same as everything else. It's just that those fungal communities were very different. And I remember when I got this data, I was feeling pretty disappointed because everything was the same and there wasn't any change. And then when I got this data off, I called Stuart, I think at home, and I was like, oh my gosh, this worked. There's a difference. Yeah.
So does that mean that we need to add that stuff every year, every 2 or 3 years?
For this sugar, that had an effect, but when we talked about soil health and kind of looking at what those communities would be, one of them is being diverse. So this might be more diverse, but it might not. It might be less diverse. And I think I'd have to look back at the data for that to see specifically on diversity how it changed. It's different from the others. But if it had shaped these communities into more pathogenic fungal populations, then this would not be something that you'd want to do. So it depends not just that it shaped them or it changed them, but we want to make sure that it changes it in a positive way.
So now that I understand the graph, can you go back to the next one?
Yeah. The bacteria. So this one, for the statistics, nothing was really distinguishable or different. They were all kind of clustered the same.
Okay.
There were a few, I think, that were slightly different from some of the treatments, but generally overall, all of these treatments were not distinct from one another.
Okay, thanks.
Mm. And Greg?
So, when we were at the Direct Seed Conference, you were in the meeting where Doug Poole talked about what he's doing with elixirs and teas.
Mhm.
And they had quite a bit on the fact that in order to get effects, uh, that you needed to have molasses in those, and I think primarily because of the sugar, so that the microbes would have something to eat in between application and where they got a life form that they were working with. So I guess in talking with him later, the fact that he was only putting on 10 pounds of nitrogen and equaling the crop yields of others that were using a full commercial package in 40 bushel land, I kind of wonder if that sugar might not be having some kind of an effect on some of the microbes, or maybe it's not the microbes, but maybe it's the phage, or maybe it's the fungal activity. But it's pretty amazing that in normal rainfall, he can uh, duplicate people buying conventional inputs without very many purchased inputs.
Yeah, that's interesting. So on the compost tea, I've been getting, I guess it's the compost extract. I've been getting a little bit more interested in that. And one of the things I think, and I have someone's compost in our fridge right now, is when you have that compost extract and you're taking that compost and you're rinsing it and pulling everything off or what you can, and you dump it on the soil. Coming back to this graph, what I presume you'd be dumping on the soil also are just a bunch of enzymes. So these are extracellular. And so as those enzymes are being sprayed down, I guess I'm a visual person. I imagine, the sprayer going, you're putting the compost tea down, and just this massive chemical reaction of unlocking and breaking down that organic matter. So that's been one thing I've been interested in on some of those practices is what are you doing on that nutrient cycling as well? So you have the microbes that would come off in the tea, you have the nutrients that are coming off as well on the extract. And then how much of these enzymes are you also putting out there on the soil? But yeah, that was really interesting, I think, on what they were doing.
Yeah, I think he was primarily into extracts, but what I thought was so interesting is the first 3 years that he started that, he told me afterwards, he had been putting on chicken manure, but then he was able to quit that and still
Mhm.
maintain the result with the extracts going forward. So
Yeah.
that's pretty interesting.
Yeah.
Well, I think what their idea is, rather than soil effect, it's more the plant takes it in and then produces exudates in order to
Mhm.
stimulate whatever's in the soil.
Yeah, because you get more of that growth. Even if it's just the nitrogen, you're putting more of that back into the soil as well. Mhm.
Kate, I've got a question for you. Thanks for the great presentation. In the regenerative space, we're seeing a lot of molasses add-ins as
Mhm.
part of a seed care treatment. And we've got a couple trials going with those treatments this season. But I guess just was wondering your thoughts on that and maybe helping me understand kind of the rationale behind putting molasses in as a seed care.
I'm not sure. I heard that from one of our producers where they're putting sugar even in on their herbicide applications. I'm not up on that for the seed codes. I think it's something to look at, especially with this data and the sucrose. But I'm not sure what the basis for that is. But that's a really good question.
Oh, thank you. We'll be sure to share whatever we, are able to figure out this season.
Yeah.
Thanks.
Mhm.
Well, do you want to talk to the grower that's doing it?
Yeah.
So the idea is, and you kind of hit on it there with the sugar, is partly just feeding microbes all the time.
Mhm.
And the other part within crop is to alleviate stress on that crop when we hit it with a chemical.
Yeah.
So I'm looking at two things. I'm trying to feed microbes, but also feed the crop and alleviate stress. The question I got for you though is on the sugar, what was that table sugar or molasses that you guys used and how did they apply it?
It was sucrose, so I'm not sure the exact form that it was, if it was like over-the-counter sucrose or if he purchased like a high-quality sugar, but it was just applied to the surface and it was added to the same amount of carbon as all of the other amendments. But it wasn't mixed in at all.
Okay.
Yeah.
Yeah,
Oh, sorry, go ahead, Hera.
it was just sugar powder type.
Like table sugar?
Yeah,
That sounds like a lot of sugar. Is it? What was the poundage, I guess, per acre of sugar?
I'd have to look at the paper based on carbon, but here you can see I think this one's the paper here.
yeah, those are paper, the white one. The sugar just blended in faster than the others, and we didn't expect much out of it, but the analysis showed different than what we expected.
Mhm.
Yeah, can you give a little estimate on the rate of sugar applied? You're looking at, looks like hundreds and hundreds of pounds of sugar that you're adding, and I'm just guessing at that.
Um,
I don't recall.
I'm looking it up for you, Dave. I have it on my other screen. So give me just a couple seconds. And okay, so okay, they had a target rate of 250 grams of carbon per meter squared. And it doesn't look like it has specifically, I think I'd have to go through it to find exactly how much was added. But for sugar, it looks like 1.2 kilograms per meter squared. That's just based on carbon. I'll look at that paper and get back to you, Dave, on how many pounds, if it's in there.
Yeah, it sounds like tons actually, looking at it per acre. So think of that in relation to what most growers are using sugar as in terms of rates. It's pretty different, I suspect.
Yeah, that's correct. So this wouldn't be like you should add sugar. So I think within the context of this, it's showing that there is an effect, but I think that was significant applications on everything. But that's a really good point. And then also with the different types of sugar being added as well. I was going to ask Chris, are you doing the seed coating yourself? So we do research on Chris Roush's field also, and I think that he has some really interesting and exciting microbiology. His soils have about half the organic matter that we have in our replicated field trial here by the site. And he has like 1.5 times the activity in nutrient cycling activity. So we're excited to look at his soils a little bit more and see what's driving that enhanced activity and what kind of fun stuff he has going on his fields. He's down in 9 inches of rainfall.
Kate, you have few questions or comments?
Okay.
The comments.
Oh, in the chat. Okay, thank you.
Mhm.
Okay. So for Francisco, how do living roots influence the formation and long-term stabilization of soil carbon? And which root-driven processes, exudation, microbial priming, or aggregate formation do you consider most important in determining whether carbon is retained or lost? So this is a great question, or a question that really is reflected in the studies that came out of this study. It was roots in the ground that had the most effective, or I guess the most durable effect on the soil carbon, is making sure you have that exudation and feeding those roots. And then also what would have been the ground cover with that. Stuart had done a follow-up 12 years after this one, also looking at those nutrients. So that's something that I can share with you as well. And another question, do those NRCS measurements include the context of the NRCS Regenerative Ag Pilot Program? The program requires participants to agree to perform soil health testing in the first and last year of the contract to establish the starting baseline and record the resulting changes. I am not familiar with that pilot program. Is there someone online who's more familiar, or from NRCS? Oh, Dave, you're
Dave,
muted.
we see you talking, but you're still on mute. Canable.
Is this the one that was just started or announced?
Yep.
Yeah, I don't know if anybody's had a chance to really get that all implemented. I mean, maybe it was just a few months ago, so that would have been late season. So I think we can watch this space for, and see how that develops. I think it's really an exciting initiative.
Yep.
So
I think sorry. I think the latest news I've heard was just they've established a kind of a suite of measurements that are going to be used. There's about 7 or so. Plus, the one that they're using for microbial activity, Kate, is PLFA, is one that they're now kind of settling on from the standpoint of moving forward. But I think here they're reaching out and asking for help from probably you and maybe others about interpretation of the testing itself. So yeah, keep your eyes open and ears open to reach out to some of the NRCS folks, because I think they're going to be wondering how to best to apply the best science to the kinds of measurements they're taking and their interpretation. And a lot of it is, like I always suggest, I think it's the kind of the before and after kind of tests that they're looking at changes, probably similar to what you were expressing with that relative difference over time.
Thank you.
So Dan, you had asked about maximizing living roots. Does it mean going perennial crops or relate to annual crops? I had just looked at what those 4 things that they were recommending. I'm on their website right now. So continual live plant and root. They talk about grasslands and cover crops in this. So I think on improving your soil health, it also has to be done within context. If you're growing a perennial, and you're in really low rainfall, is that something that you can achieve? And then also, is it marketable? So thinking about it within your cropping system and your region, and doing the best that you can do.
Okay, just a related question. That if you maximize living roots, even if you have an annual crop, you're going to take a hit on soil water. So is there any negative effect on beneficial microbes? Yeah, if you're losing water by intensifying?
And this data here, this is the only time that I really compared the annual versus that, and with those durable effects, and when wheat was cropped. So That definitely was different than those positive effects that we saw on the nutrient cycling enzymes. When it was that perennial crop for those 5 years, it had those lasting effects. So annual versus perennial had a big effect there. And that data is right here, where we can see the annual crop. So whether it was fallow, brassica, or wheat, these two annual crops, they did not retain that nutrient cycling activity. So there would be an effect, but just like you said, with that cropping system and how that works. Does that answer your question, Dan?
Yeah, thank you very much, Kate.
Okay. And this is a great question by Dave Huggins. Challenges relating structure to function, any insights? So that I think is a really big question, and where the microbiome comes in is how do you pull this apart? We looked at DNA as our estimate for a biological abundance, and it's not a perfect estimate. Some of the data that's used is phospholipid fatty acids, as you said, also the fatty acid methyl esters to try to look at abundance, but really teasing out that structure to function and that nutrient cycling. To me, I like to look at both sides. I like to have those functional measurements. I like to look at some of the gene copy estimates so we can target the functional genes, especially, with the nitrogen cycle, where it's difficult to measure some of those nitrogen transformations. But I think when it comes to the genomics or the metagenomics and then really tying that straight back, I think that there's a lot of roadways and successes going that direction, but I think it's definitely a challenge and things to overcome. And I know that there's a lot of work, kind of trying to pull those apart right now. Was that feasible, Dave? Okay.
Yep, no, it's always something that you're wondering about, right? In terms of particularly with the definition of soil health and trying to, maintain function or improve function, and then trying to relate a lot of our measurements in science to how that is, achieving some of the kinds of function expectations we have for soil. So I think it's a huge challenge for us, and it's complicated. So
Yeah.
I was just seeing if you had some insights. You could probably talk for the rest of the day on it, I suspect.
Yeah, it's funny because even on on the estimates we do for enzyme activity, Veronica Costa Martinez is also on this call. Sorry you're going to hear this, Veronica. I talk to her and call her the grandmother, mother of soil enzymes, but godmother, that's what it is. But on the soil enzymes too, when we measure those, we measure them in conditions that they're optimal activity too, which is not necessarily reflective of what's going on in that soil and that ongoing activity and those changes. Yeah. So we have a lot of good challenges to overcome. That's my optimism. Elliot asked or posted that they'll be utilizing the updated conservation evaluation and monitoring activities, SEMA 216 is the mechanism for testing. She's also posted a link for the training on that if you look at the chat box. Okay, so I guess we have 4 minutes before we're going to get booted by Zoom. I want to thank you all very much for coming. If you have questions or if you're dying to be a speaker for next year, please let me know. I have 4 spots available right now, so get your name in early. And I want to also thank thank Oregon Wheat for hosting this platform for us today and for the collaboration as well, and along with our OSU collaborators. So I hope that you join us again next week. Dave, you look like you have something.
Yeah, Kate, is there a field day planned for out there in Pendleton, maybe later on in the summer or fall?
Yes.
It might be farther out.
Yeah, That would be great. We have field day coming up in June, so
Okay.
I'll send you that and we'd love to have you out. Dave is our national program leader once retired.
Yeah.
Yeah.
I'll share that with Dan Roberts too and others. So yeah.
Okay.
Thank you. Good.
Okay.
Great. Thank you.
Well, thank you, everyone. I really appreciate you joining us today, and I hope to see you all next week. Same time, same place. Okay. Thank you.